Good afternoon. I think we are already enough to start. So, plan for today. Original plan was to talk about UMAP because that's the big outstanding thing we always look at with UMAP plots, and I haven't yet explained to you how to do that, but I didn't get this finished to prepare. So I postponed this to the next and last lecture. And for today, I wanted to show you another way to do pseudo-time. We already have one way. We will see later why we have two and what the difference is. But another reason why I like to show that is that it's actually quite beautiful math. And I guess maybe some of you can appreciate that one. So maybe to first point that out. So if we have cells which are all very similar, and then we have the cells which make a rather big jumps, and then we again have similar cells. Then if we do a principal curve, then usually it's easy to have a principal curve going through here, and then a lot of pseudo time will pass from here to here, which one may or may not be good. We might say that it's good that the pseudo time evolves slowly here, and then here it evolves fast, and then here slow, because we imagine this means that a lot of cells are in this state, and then they rapidly change to that state, and during that they go through this. But because we measure the Euclidean distance through feature space, we don't see how things speed up here, because we don't see that there's much fewer cells. And this can cause changes in the order of magnitude of these cells, of how quickly these cells move by many orders of magnitude, because sometimes the density is much smaller, much higher than elsewhere. I think at some point we have done this UMAP where we colored each cell according to the distance to all their neighbors. Then there is another idea which is quite popular. And this is, we just take the neighbor graph, and now we do a hopping on the neighbor graph. That means we do a diffusion process where we start somewhere, and then we imagine at each time step, we hop from one cell to one of its 20 nearest neighbors. And then from there, we hop to the next cell, and so on. And then we see how quickly we reach the other half, and that will be a distance to a measure of a distance. And this is something which you have in a lot of different areas of applied mathematics. We call it diffusion on the graph. And why we call it like this is, let's maybe start with this. Imagine usually diffusion processes means you have something which slowly spreads out. The classical example of a diffusion process is you have a piece of filter paper and you put a spot of ink onto it, and then you see how it slowly spreads out. And typically you will find that this spreading out goes with a square root, that the radius of a spot that you see on your filter paper scales with a square root of time that have elapsed. To whom does this sound familiar? Have you ever done diffusion? Yes. You might, and there's two ways how you call this. One is the random walk, where you just imagine a ink particle sits somewhere at the beginning, in the middle, and when this ink particle at each time, after each time point, it makes one step in a random direction. And because it makes steps in a random direction, it might move out, it might move back, and so on. And this is, of course, precisely what you have in a Brownian movement. Whenever you put a drop of ink somewhere, that's simply due to thermal fluctuations. The ink particles are pushed around, and you see them performing this random walk, which is completely indirected. So from every time point to the next time point, the directions are uncorrelated. And typically, if you look at this in 1D, then you might say, if this is our position and this is time, you start somewhere in the middle. When you go maybe a step up, a step down, it can be anything like this. And if you make many such trajectories, then this will be you will find that the probability of ending up at a certain point at a certain time is always a Gaussian. And the width of this Gaussian is the square root of the elapsed time. And that's, at least in physics, a pretty fundamental thing. As long, whenever you have a directed movement, then of course you always make a step in the same direction, so you move with constant velocity, and the distance that you have, that you move within a given time is proportional to the time. If you wait twice as long, you have moved twice as fast. But if it's a random walk, where at every time point you decide at random whether you move forwards or backwards, the amount of distance you will cover is expected to be only the square root of the elapsed time. And this is a rather, well, it appears in many things, and here we use the same. Only we allow our walker to not only walk forwards or backwards, but we put the walker on a random point in our graph and let him always hop from one graph node to a neighboring node. And we call this then diffusion or random walk on the graph, where if you sit on it, yes, exactly, you simply always make a step from one vertex to a randomly chosen neighboring vertex. And each neighboring vertex has the same chance of being chosen. We'll see in a moment that there's two ways. You can either describe this as a discrete process with discrete time steps, and in each time step you walk from one vertex to a neighbor, or you use it as a continuous process, which is of course more appropriate if you want to understand this spreading of ink. And as usual in physics, you can start with a discrete picture and then take the limit to arbitrarily small time steps and arbitrary small spatial steps, and then you will find again the continuous limit. But let's start with a discrete time step, and I show you how this looks like. I should probably simply load it here as well. SCO20, they've selected. Maybe I should leave the door open. I think it's strangely hot here. My office is nicely cool. So, am I now here, and where is it? Graph diffusion. So, the way it works, and you can also read along on the web page, I read, well, just my usual standard example data set that I always use, and I run now a standard server analysis, which now runs for a minute, and while it does that, we'll quickly have a look of what we'll do next. And I want to always switch from each cell to its nearest neighbor. For this, I have to, of course, find the nearest neighbors in PCA space, and I do it as we did it already a while ago. I take the PCA coordinates for each cell and use this functional nearest neighbor package to get the 50 nearest neighbor. And then it gives us here a matrix like this, which you can read as the first vertex or the first cell has its nearest neighbors, the cells 2504, 4613, 2817, and so on. So each row shows you the nearest neighbors of the first, second, third, and so on cell. And if we want to consider, if you want to consider this as a graph, we can construct an adjacency matrix. And just to see here how to do this adjacency matrix, if, for example, I want to connect each cell with its third nearest neighbor, and I simply take the third column out of this nearest neighbor list and write this third column next to simply a list from 1 to 10. When I see the third nearest neighbor of 1 is that one, and of the third is this one. And of course, if I now let here this go through all the columns, where I take each of these columns, then I get the complete adjacency list, so a list of all the edges I have in my graph. And I can do this to initialize a sparse matrix, where I say make a sparse matrix where this here is where you have always a 1 in all. Remember, I want to make an adjacency matrix, so a matrix where every column, every row corresponds to a cell, and if the cells are neighbors, there's a 1 in the corresponding entry. And so I first make a matrix where there's a 1 whenever the cells are nearest neighbors. So I take here the list of all the cells, and here the list of all their first nearest neighbors, if I is 1, and everywhere I put a 1, and then I get a sparse matrix with these 1s. And then I do the same for the second, third, fourth, up to the 15th nearest neighbors, and I add up all these matrices. This is simply a way of using our sparse matrix function, because adding it up just means that it adds up all these 1 position, and in the end I get a matrix where there's 1s from every cell to every of its 15th nearest neighbors. See if it has finished this here. Yes. So here is my Hume plot. Now I can also show you this. I make my nearest neighbor plot that I've just done here. And now here I can here, for example, use different values for I, like this is the first nearest neighbor, second nearest neighbor, and so on. And then I make here this sparse matrix, which now, as you can see, is a matrix which has in this rows, in this columns, this 1. And in that way, I now get all the respective. Which way have I done this way? Have I written it this way around? Oh, it must have somehow reordered my thing in some way, so that it's sorted by J and not by I. Doesn't matter. And now if I add up all these matrices, then I get a final matrix with now all adjacency, with now all edges of my neighborhood graph written here. And the last thing that I've done here is that I add to this matrix its transpose. Because you remember sometimes it happens that a matrix that cell I considers cell J as the nearest neighbor, but not vice versa. And this gives us a directed graph. But if I add the transpose, then I also add all the edges where start and end are swapped. And because in my adjacency matrix, the rows are the starts and the columns are the end, and if I transpose it, then I get the opposite. So I can just add to the matrix its adjacency matrix and its transposed matrix, and then I get the symmetric graph. Except for the little point that now if I look at this matrix, it contains not only 1s but also 2s. Because maybe an edge really did appear, 2 cells considered each other mutually as nearest neighbors, when I already had a 1 in the upper and in the lower diagonal. And if I now add them up, I get a 2. But that's easily rectified. I just have to put all the 2's back to 1. Essentially, I want that all non-zero values are 1, and this is why I use this here and tell it x, so the values should be all the 1s. And now I have this adjacency matrix, which you see encodes that from all these nodes to all those nodes. Oh, damn it! Should be like this. It's switched from a coordinate sparse to a column sparse matrix, so it's not as easy to see anymore. But it's still, the matrix should be that way, that every one we see in here tells us an edge. The reason why I want to use a matrix is because that makes it very easy to write as a random walk. What I now do is here A is our adjacency matrix with 1 for every connection. And what I now want to be able to write is something like, I put this here. No, and I write it on the board. Thing like if, for example, I have my adjacency matrix A here, and I now read Aij is whether there's a vertex from I to J. And if I now come in here with say, basis vector, the ith coordinate vector, which has a 1 at I and 0 everywhere else, and multiply with this matrix, then of course this will select the ith row from my matrix A. And the ith row contains exactly ones at the neighbors of this thing. And in that case, I can use this now as a transition matrix, because what I want, you might already have guessed if you know the term, that we have a Markov process here. In a Markov process, we have a system which is in a certain state, namely the walker is at a certain place, and every time step we have a transition. And the transition only depends on the current state and not on the previous states. And if we have this thing, then we can now write the current state as a vector, which tells us for each vertex the probability that the walker is at this vertex. And we start off with putting the probability, putting the walker at some place, EI, which we say is, it means we put the walker on vertex I, and then after one time step, the walker will be at any of the places which have a 1 here in the ith row of A. Obviously, this is not this does not mean this is not a probability anymore, because if this vertex I has 5 neighbors, then we have now 5 ones in our thing. But we would want that it tells us with a probability of 1/5 it goes to each way. So we want the rows to add up to 1. If the vertex has degree 5, then each element in this thing should be 1/5. And that's what I've written here. I take this adjacency matrix and divide each row by its sum. So this is the sum over all the rows. And afterwards, I get what I call the transition matrix. And the transition matrix tells me the probability that the walker will end up on vertex J if it was before at vertex I. So, and this is the central way how you always work with Markov processes. You have a state vector which tells you how likely it is that the thing is that the walker is at each position, and you have a transition matrix which tells you what's the probability of going from position I to position J. And the nice thing about this thing is if you go in there with a unit vector like this, which has a basis vector which has a 1 at a fixed point, indicating that the walker is with probability 1 at vertex I, then afterwards, after you pull it through A, you get a vector which tells you the probabilities where it is, and it has something like, say, 1/5 on all of the 5 neighbors of the vertex I. You might notice here, I've written it here from the left. Normally when we write matrix vector multiplication, we write it from we write the matrix followed by the vector. The Markov people do it the other way around, because they started off with saying the matrix Tij has elements which tell us the probability to transition from I to J. So Tij is the probability if you're at I to get to J. And if you define it like that, then you have to say one row of T tells you all the probability vector that you get if you start at I. And hence you need to do calculate your transition by multiplying the current vector from the left on it, so that afterwards you get it out. Which is, I wondered a bit whether I should turn it around to the usual thing, but in the end I stuck to this because that's how it's most textbooks on Markov. And so with that, we can now easily try this out. So here first I have to take my adjacency matrix and turn it into a transition matrix. And if I now check the sums of this transition matrix, you see they are all 1. Before, if I take the row sums of the adjacency matrix, then each one tells me the degree of this vertex, how many neighbors it has. And you remember we started with 50 neighbors, but because we symmetrized, each node can get additional neighbors. And and now afterwards, if we now have this symmetrized here, they are all 1, and of course the individual things will be something like 1/17 or 1/18 or whatever we have here. And if I now start with and exactly, we can now try this here. I want to try it with one specific point, and I wanted to have a cell which in the UMAP sits somewhere around here. You see around a UMAP coordinate of 1 and 1 or so, or 2 and 1. So I decided to simply pick such a cell by saying, take the UMAP coordinates and find the cell which is closest to point 1, 2, by calculating here for each cell the distance to the point 1, 2 in the UMAP and finding the minimum, and it told me it's this cell here, the cell number 16,000. And that's the black cell here, which is just to pick a random one which sits about in the middle. That was the whole point of this. And the purple one are its neighbors. And what I've now done, of course, is I make it black if it's the cell that I've just selected, and I make it purple if it's if the adjacency matrix has a 1 next to this in the row corresponding to this cell. And with that, I've calculated it. And now we want to do 10 steps. And so I make 10 such hops. And what I do, I start off with a vector which has a 1 at position I, and all the other positions are 0. So this is like a sparse matrix, but now a sparse vector. That means everything which is not explicitly mentioned is a 0. So in this sparse vector, I can multiply from the left now with my transition matrix. And then, of course, I get a useful result. So let's do this. I start here with this vector first. I haven't selected my cell yet. I select my cell, I make the plot, and now I can take here the sparse vector. If I look at it, it gives me this long vector and tells me here what's in there, but I can also look into it like this, and it tells me there is exactly 1, 1 at position 16046. And if I now multiply this vector from the left with my transition matrix, then I get a new vector, and I can look at this, and you see the new vector now contains it contains values 0.05 at all these 20 positions. And if I do this again, I take this thing here now and multiply it again with TRM, then I get now already 102 non-zero values. In that way, I can calculate, for example, 10 different positions, and always my vector will end up still being normalized and having the sum 1, because that's how we constructed our transition matrix, that it makes sure that it always gives us gives us a valid probability vector. A valid probability vector is a vector which only contains values between 0 and 1, and that sums up to 1. So that we can say with 100% probability, one of these elements describes what we have. So this is what I've now done. I multiply from the left this basis vector, which has a 1 at I, and multiply 10 times the matrix T from the right, the transition matrix. And if I do that, then I get this here. And here you now see this typical usual random walk thing that you also see from a diffusion. Imagine this whole thing would be a yellow thing, would be a piece of tissue or filter paper and so on, and you put a blot of ink. It would probably look like this after a while, with most of the ink still sitting in the middle and it's slowly fading out. And what I want to show afterwards, just that you connect it to the physical picture, is we make a graph which is a grid. And if we make such a grid and start somewhere there and then calculate this thing, we will also see the diffusion like an ink plot which slowly moves out. And actually, let's quickly do this because I had prepared it. Where have I put this here? Graph diffusion. No, where was it? Graph spectra here. So what I do here is I make a 30 by 30 matrix, And what I've now done is, as you can see here, I add in my adjacency matrix, I always make connections from the point ij to the point ij plus 1. That's all these horizontal connections. And then I do the same for the vertical connections. And here I go the rows from 1 to 30 and the columns only from 1 to 31 to 29 because I have only 29 horizontal connections. And the same for the vertical connections here. And I already symmetrize it here. And now with this adjacency matrix, I can again start with some vector. Let's say U is, what's happened now? This is a brand new computer. Well, not brand new, it is what it used, but still. Come in. Try this once more. Okay, probably I have to. So, repeat 0, L squared. So this is now a vector with all u's. And the funny thing about this vector, if I make it as this vector u, the components correspond now to these grid elements, 1, 2, 3, 4, up to 29, or 30, 31, 32, 33, and so on. That's how I arranged it. You probably would have to look at this code a bit in more detail to see that this really works out. But what you can believe me, the adjacency, we have 30 squared vertices, and the adjacency matrix has 900 rows and 900 columns, and the 900 rows correspond to this thing in the 900 columns as well. And that means that a vector of 900 rows or columns, I can plot it, and it's all 0, but of course they are not next to each other, they form a grid. So what I do is I make a matrix, I tell it, make this a matrix. And now we get a matrix like this, and I tell it, plot this matrix. And now, well, it's a matrix of zeros. Now I set a random one of these to 1. Let's say 900, 420 to 1. That was a bad one because it's at the corner. So here is something now for 1, and now I can take this matrix and make my transition matrix. Is the adjacency matrix divided by the columns of the adjacency matrix. Now the row sums of the transition matrix should be 1. And if I now multiply my U with this transition matrix, then you see it spreads out a bit. If I multiply it twice with this transition matrix, it spreads out even more. Here we have slight problems because actually something is not right here, because why does it always jump over 2 of them? No, that doesn't work properly because it always no, I think that is fine. It's only because I did it a way that it always has to jump. So whenever it sits somewhere, it jumps to one of its neighbors, and this is why we have this checkerboard pattern. But you can see how it slowly spreads out more and more with each time I do this here. So this is really a typical diffusion, the same way as you usually do it with if you have some ink drop or so, which then slowly spreads out. You can also, if I now looked at the profile of this, I would get back to the Gaussian. Now let's go back here to to this thing that I've done, to our usual thing, where I've said I start. Now I've messed up my transition matrix by making this adjacency matrix. So now it's correct again. Bosoms, find a cell, plot the cell with its nearest neighbors. And now here I now multiply it 10 times and plot this again. And this time we now see this is spread out after 10 steps. And now I want to see how it's after 20, 30, 100 steps. And you can already see what I'm doing here with this for loop. This is very clumsy. I do this, and if I want to do 1,000 steps, I have to do 1,000 vector matrix multiplications. It would be much easier if I could simply take this matrix T to the 1,000 and then multiply the vector only once with this matrix. But is it easy to take a matrix to the 1,000th power? And actually it is. Namely, it becomes easy if a matrix can be diagonalized, because if you can make an eigendecomposition, then it works very easily. And I can show you how this works here. So here, let's now exponentiate it. We want to take the 11th power in order to simulate L steps. And here, what I first do is I make this matrix here, which I call the degree matrix. The degree matrix, as you can see, is a diagonal matrix, and in each diagonal element, it tells me the degree of this vertex. I've written it a bit strangely here. You see this here means add up all the values within row I, and this here means write this element not in everywhere in row I, but only in the element where I and j is the same. That's a Kronecker delta. I don't know if you guys know that, but it's just a notation for diagonal matrices. So now we have this matrix, and the diagonal, it has the degrees, and if we take d to the -1, the inverse of a diagonal matrix is simply the reciprocal of the diagonal. So d to the -1 is 1 over the degree all over. And if we multiply a diagonal matrix from the left with a matrix, it scales the rows. So what this thing here does is exactly the same as we did before, normalizing the matrix such that the row sums are 1. Because each element in D is 1 over a row sum, and multiplying it with a row gives the normalized matrix, the transition matrix, which I said, remember the adjacency matrix is symmetric because we have an undirected graph, but the transition matrix is no longer symmetric because we made sure that the rows are now add up to 1, and that of course messes up the normalization of the columns. And now, as we've seen before, if I go in with a walker starting at vertex I and I do one step, we have to multiply this from the left to pick out the ith row of T. And if I do this L times, then I have to multiply it from the left and multiply T L times from the right. So I have the 11th power of T. And in order to calculate this, now you can do neat eigendecomposition. It works as this. We first, you might remember there is the rule, you know, the principal theorem of matrix calculus, that every real symmetric matrix can be diagonalized with real eigenvalues. So, but as I just said, the matrix T is no longer symmetric. A was symmetric, T isn't. Can make a compromise, which is sort of half normalized and half symmetric. Instead of multiplying d minus 1 from the left, I multiplied d minus a half from the left and d minus a half from the right. So I divide each row by the square root of the vertices and each column by the square root of the vertices. So the element ij is now divided by the square root of a vertex, of a degree of vertex I times the degree of vertex j. This is a matrix which we now no longer quite understand what it means. It's neither an adjacency matrix nor is it a transition matrix, but it is symmetric, so we can find an eigendecomposition. We can find eigenvalues lambda, and we can find eigenvectors, which are the columns of U. And as you know, whenever you have found the eigenvalues and the eigenvectors, you make this matrix U out of it, and then you can write this matrix as U lambda U transposed with lambda diagonal. And now we can use this to calculate T to the L, because T to the L is, as we said, T is D to the minus 1 over A, so the normalized A. And the D to the minus 1 I now write as D to the minus a half times D to the minus a half. You see these two together, they are D to the minus 1. And on the right-hand side, I have D to the minus a half times D to 1 half, which gives, of course, the identity, because minus and plus and halves cancel out, and this matches the fact that here to the right of the A we have nothing. And if you now imagine you write out this parenthesis, you write L times next to each other, D to the minus a half, A, D to the minus 1 half, then you will see that what you get is, let's quickly write it down, then it's clearer. We can write this as D to the minus 1 half, and then we have D to the minus 1 half, A, D to the minus 1 half, then again D to the minus 1 half, A, D to the minus 1 half. So this would be 1, this would be 2, and so this L times. And finally D to the 1 half. And of course, this is the same as D to the A to the L, because this here gives a D, and here we always get an A, dA, 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 and at the end we are left with this D to the minus 1 half, D to the 1 half. That is this. So this shows you how this first equation, saying this one here, works. But the next one I can now do is what I've written here in the parentheses. That's of course T tilde. And for T tilde, we've just said because it's symmetric, we can write it in its eigendecomposition. So I replace this thing here with that one using this. And now I can easily write it because this UT, when you write this out, you always get U lambda UT, U lambda UT, and this L times. And in the middle, you always have UTU, and UTU is, of course, the identity because it's a rotation matrix. And this, in the end, is the whole point of why people do eigendecompositions. Eigendecompositions allow you to exponentiate a matrix, because if you have an eigendecomposition like UDUT, and you take it to the 11th power, when this is the same, I shouldn't call it D, I should call it lambda. When this is the same as U lambda to the L UT, because you can pull the exponent inside to the L, because the powers from the UT times U, they always cancel to a 1. This only works because UT is the identity. And the advantage here is a diagonal matrix can be exponentiated very easily, because a diagonal matrix, you exponentiate it by exponentiate each value separately. And so this is the general point. Whenever you want to exponentiate a matrix, you diagonalize it, and then you use this identity for the diagonalization to only exponentiate the eigenvalues, which stand in lambda, and then multiply the eigenvectors from both sides around it, and then you get the matrix exponential. And this now allows us to get this T to the L. And I've already underlined here a part which we later will have to call XL. But let's first try this out. So first I make this matrix D here, which I simply call degree diagonal. And you see this is a sparse matrix where we have only elements at 1, 2, 3, 4, at row 1, 2, 3, 4, column 1, 2, 3, 4, so only at the diagonal, because row and color coordinates are always the same, and the values are the degrees of the matrix, so the row sums of the adjacency matrix. And now that I have this, I can also write down the inverse degree diagonal matrix where we have 1 over the degrees, this thing here where we have the square root of the degrees, and this thing here where we have 1 over the square root of the degrees. And now next I may have to make my eigen decomposition. So I take this t tilde, which is d to the minus 1/2 t d to the minus 1/2. So d to the minus 1/2 inverse square root dec dx times the adjacency matrix times again d to the minus 1/2. And I tell it I want the eigensystem of that. And I'm happy with the top 100 eigenvectors because otherwise it takes too long. And with this, we can now calculate ei to the tl as follows. We calculate first this part here, xl, which is d to the minus 1/2 times u are the eigenvectors, times the diagonal matrix with the eigenvalues, and now to the 11th power. Here I said this is for L equals 10. And then what I get out of that, somehow R doesn't automatically make a matrix, and then we get this thing which I call x10. And I have to multiply the x10 then with this part here to get my actual stuff. And hey, this is not what I wanted to show you here. Let me quickly do this and fix the mistake I just found. So here you see this now takes a while because this is a big matrix, but it's sparse. And now here you might say this isn't correct because I didn't use the whole eigenvectors, I only used the first 100 eigenvectors. But luckily, if I take these eigenvalues here and plot them, you see the eigenvalues themselves, there's lots of eigenvalues, and even the 100th eigenvalue is still quite big. Should maybe write here y-lim is 0,10, so that you can, no, 0,1, so that you can see the eigenvalues go down slowly. But if we take the 10th power, then it looks much better because point I to the 10th power is already 0.2. So we might say that probably using only the first 100 eigenvalues is okay because all the later eigenvectors will be multiplied with rather small values. If we go even to L equals 100, you see it seems to be clear that 20 eigenvectors or so should be, 20 or 30 eigenvectors should be sufficient. And with this, we can now easily calculate what we've done before. I multiply this x10 with the rest, with the eigenvectors transposed with the other one in order to complete here this formula. And with that, I can now plot the thing, and as you can see, it looks the same as before. This here was where we manually took the matrix, took the vector with the 1 here at this cell and zeros elsewhere, and multiplied it 10 times with the transition matrix, and here we now used our calculation over the eigensystem. And that, of course, is less precise because we didn't use all eigenvectors, but only the first 100. But here, this is the exact calculation. This is here using only the top 100 eigenvectors. And each point is the probability that we add up at a specific cell. So it's a color value of one of these points here. And as you see, it agrees well. The point of this whole thing is now, first, you can easily now, of course, try different calculations and quickly see how this thing spreads out more and more. Let's quickly do this. Where am I here? So I have this x10 is what I calculated here. And I put this down here, and then I get u10. And here the whole thing now gives me this matrix, this thing here. And now we can quickly see what happens if instead of a 10, I take 20 or 50 or 100 or maybe even 1,000.
And you see how now, after 1,000 steps, it's basically spread out everywhere. And of course, if we take very many steps, it will spread equally everywhere, and we have the same, more or less the same value at all places. Not quite, because the cells with higher degree will have a higher equilibrium distribution than the cells with lower degree. That's why it still looks a bit speckled. And also here, this thing seems to be completely disconnected in our graph, so it never gets any cells if we start here. There seems to be no edges from these cells to the yellow cells here. But overall, you can see how this diffusion works. And what we can now do is we now define a diffusion distance. And the idea of a diffusion distance is we fix a number of steps. We say we want to get the diffusion distance for 100 steps. And then I might start this random walk here, and I start another random walk here. And then I ask how much do the distributions around this random walk and this random walk, how much do they overlap? And this is what I call the diffusion distance for 100 steps. You can see if you take, for example, and this diffusion distance works very different than Euclidean distance. Imagine we make a diffusion distance with only 10 steps. Then any two cells which are further apart from that one will have distance infinity, more or less, because they have no overlap. So it sort of shows you only whether it's neighbor or not at all. While if we do it like this 100, then it means everything which overlaps over this stage is considered near, and everything which overlaps at the distance, which is now yellow, maybe this cell here and that cell here. If I started a walk from that cell, it would go only here. Then I would get no overlap because there's a big yellow gap in between. Yeah.
How is this overlap formally calculated?
We come to this at the moment. Simply the Euclidean distance between the two probability vectors. And with this, but the other point is that if there's really tiny differences, for example, you see here this substructure that we can either go this way around or that way around. This will be completely averaged out by the diffusion because the diffusion has flown around it. So by choosing the number of steps, we say the distance scale at which we are interested. We say, tell us whether the cells are far or near on this scale. Everything which is much smaller than this scale, simply don't care and average it out. Everything which is much larger than this scale, say this is simply infinitely far away. And that can be useful because then you can have different distance scales which look at a different level of detail. Yeah?
It wouldn't be infinite, right? It would be very finite.
Yes, nearly infinite.
No,
Very. Yes, in this case, actually, yes, because imagine you have two cells where the shortest path from this cell to that cell goes over 20 hops. Then if I make less than 10 steps from either side, they don't meet at all.
So then one has a probability distribution with zeros for all those cell indices, and another those cells, another one with overlap.
Oh yes, you're right. Sorry, wrong. Exactly. There's a difference. If I was in my mind, I was at the product. But yeah, for a difference, we don't have. Exactly, it's the other way around. The distance is basically 1 because all the probability distribution sits, one part of the probability distribution sits only in the left half and the other part sits only in the right half. This part adds up to 1 and is always compared with zeros, and vice versa when I even get 2.
But that wouldn't work with the L2 norm, right? Since we're squaring all these probability limits, wouldn't we do the L1 norm?
Right, if it were L1 norm, exactly, yeah. You catch me now with having looked at this only yesterday and already forgot about this from last year. But you're perfectly right. It's basically because we use the L2 norm and not the L1 norm, it gets a bit more strange. But in the end, we want to do this qualitatively, and I show you now how we do this. So here I now calculate this diffusion distance. I calculate this diffusion distance. Let me quickly put my phone on silent. By as follows. In principle, I would calculate from what I've just told you, I would calculate it like this. I start at the cell I and make L steps and get this probability distribution. I start at cell J and make L steps and get that one. And now I compare them, say, as L2 norm. But if I want to calculate this in practice, I get a bit of a problem, because to calculate it, we have to do this here. D to the minus half U lambda to the L U T D to the 1/2. And the nice thing is this part is easily calculated, because I just have to look at the first, if I say I calculate my eigendecomposition with 100 eigenvectors, then I only have to look at 100 values here. Because all the other eigenvalues are implicitly set to 0. So I get this matrix XL, which is, of course, a matrix with 1 row per cell, but only 100 columns, because the rest has been cut off. And now I have to make this again to the big long matrix by multiplying it with UT and with a D1/2. And I don't want that, because the nice thing is that I can calculate with vectors which are nice and short, only 100 elements, even though I might have 10,000 cells. And up to here, this works because XL has only a limited number of columns, only 100 columns. But once I multiply with this, it gets blown up to the full size again. But now this is only a rotation matrix, so it doesn't change anything about the distances. So ignoring that one doesn't really matter. It matters only because we need it in order to be able to multiply with D to the 1/2. Now, what if we were to ignore the D to the 1/2 to the very right and simply say, that's probably not so different from unity, from the identity matrix. Then I can also skip the UT, because it's only a rotation, and then I can take the Euclidean distances only of the XL. And that is actually what people do, in fact. They say that the degrees are always there at least 20 and at most 40, so that doesn't, it's only a factor 2 back and forth. And with this logic, I can then say, let's calculate our distance not with t, but only with x, so only with the part up to the middle of it. And if you think about it, there's here now the ut missing, should be in here, but it basically means that our distances are just a little bit differently weighted, because we have here weighting factors, which should be spread out according to the eigenvectors with the ut, which is here missing. And with all this together, I now can say, let's simply take these distance values. And the reason why people like to do that is that now we get this big matrix XL, which, as I said, is a matrix with 1 row per cell and only 1 column per eigenvector. So each cell is described by a vector of length 100, which are the first 100, which correspond to the first 100 eigenvectors. And that now makes my calculation very easy. I simply calculate this matrix X1, X10, and in this matrix X10, I have the probability vector on the eigenvector in the eigenspace for each matrix in each row. And if I want to calculate the diffusion distance between 2 cells, I simply take the 2 rows of this matrix X to the L and look at the Euclidean distance between this. And that's what I do here. I calculate the distance of each row in X10 with the row corresponding to this cell. And now you can see this thing is now colored by the diffusion distance of every cell to the cell here at the yellow point. And you can see how this works at a level of 10 steps. It more or less resolves distances up to this space, and everything beyond is just some flat out green. And here I did it for different numbers of steps, for 3 steps. for 10 steps, for 30 steps, for 100 steps, for 300 steps, and for 1,000 steps. So you see that each choice of numbers of steps looks at a different level of details. Yeah.
Isn't the L2 norm a bit introducing artifacts because of the squared sum of probability elements? You can see that it's not uniform as you have these numbers of steps, increasing numbers of steps, which is because you just take these two squared sums of probability distributions. And so if something diffuses more, it will have a lower diffusion distance.
We can. There's a nice thing about code like this. We can simply try it out. So here, this is what I've just done here. And now you would like to use the L1 norm of this. So let's remove the square root and remove the square. And instead, put a absolute value in there. Where exactly do I have to put this now? Apply. This here is the comma 1 comma function x. No. Now I remember how to do it correctly. So I take, instead of squaring, I take the absolute value of the difference. And now I take the row sums and I don't take the square root anymore. See how it looks with this. 3 steps, 10 steps, 30, 100, 300, 1,000. Now I've lost, of course, the other one. It's no longer that easy to compare. But I see your point. That's probably up for debate whether the other norm might be more appropriate, especially as we're looking at probability distributions anyway. People might like the Euclidean norm because implicitly, of course, we use here the fact that we have this rotation with u here. Where was it? No, I don't find my, here. We take this XL and then we say this is more or less the same as the real probability distance if I do this rotation. And L2 norms are invariant under rotation. L1 norms are not. And that might be a reason why the L2 norm might be more appropriate here. So, and now we wanted a pseudo time, and the way how I now do the pseudo time is rather simple. I decide I want to get a pseudo time which goes from here to here, along here, round, 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 up to here. And for this, I have to find a starting point and an end point. For the starting point, I take this cluster here. Where was it here? The cluster 0, and ask which cell is furthest out in the cluster 0. And the way how I do this is that I ask for each cell, here at my 100 cell steps, how far away is each cell from here, that cell, which is somewhere in the middle. So I take of all the cells in this red cluster here, the one which has the largest distance. And then I should get something here, which is at the beginning. So I take all these distances, and this is the distance of the cells in the red cluster from my cell, which I've picked randomly in the middle. And as you can see, there's here a few cells which are really a bit farther away than all the others. In the end, I've decided to take this cell here because maybe this is a bit an outlier, some kind of strange tail, which sits, like you can see this part here. These last ones in the histogram might be these ones, and I want something here. So more or less arbitrarily, I said, let's take the cell here, which has this distance at 0.0013. And I find the cell which has a distance of 0.0013 to my cell in the middle and call this the start cell. And in the same way, I look for a last cell here in the cluster 7, which is also as far away as possible from this cell here, by checking again the distance of the histogram of distances from our cell in the middle to the cells in cluster 7, and then simply decide I take here the cell at the very end. And now I have a start cell somewhere here and an end cell somewhere here. And now I calculate the distance of every other cell to the start cell and to the end cell. Right, where's my start cell? Start cell and end cell. And with these two, I can now make this plot here, where each cell is a point, and the x and y coordinate tells you the distance to the end cell and the distance to the start cell. And the colors that I've used is I used green for everything here along the lineage, red for those cycling here, blue for the ones further out. And if you now look at this, maybe I try to zoom in a bit. Can I simply take this out? Yep. You can see that it really seems to bring the cells into some order. Like this seems to be the cell which is closest to the start cell, so this is the start cell. And then I have to go here, click closer and closer to the end cell, and now I have arrived at the end cell here. And these cells here, they seem to make some kind of strange shortcut, which we might want to interpret more. But in any case, we can ask the progression along this line is probably a good pseudotime. And the progression in this thing, this is easy to see, that's the difference between the end and the start cell. So I do this and plot here the difference. Is it closer to the start cell or closer to the end cell here? And here I take the sum of both of them, so how far are they away from the shortest path between them. And now we get this really nice pseudotime, which shows us, okay, we can really start here and end here. And if I now give for each cell this distance to start to distance to end, and call this my pseudotime, I now get something which starts here, increases from here to here to here, and then increases rather rapidly here. Increases here so rapidly because there's so way more possible hops on that side than here, because here the cells have lower degree. And I can also check this other thing, this y-axis, because these cells up here, they are so far away from start cell and end cell that probably the pseudotime doesn't really make sense to apply to them. And that's the cells which appear here now in blue. And we can see the red cells are probably where we can use the pseudotime, and the white and blue cells not so much. Now the last thing that I want to do is now compare this pseudotime with the principal curve that we did last time, where we said we try to find a curve through PCA space, which is as close as possible to all the red cells. And I recalculate this principal curve here, only using the cells which I marked green here. And takes a second. Takes more than a second. I know, it's gone. I can again, if all the cells which, and now here is now the principal curve according, the pseudotime according to principal curve, and I projected all the other cells also onto that. And now here I can compare the two. And here you see green is now the lineage. And as you can see, it agrees more or less, but here is a stretch where the diffusion pseudotime doesn't advance, but the principal curve pseudotime does. And that is the cyan-colored cluster, which is that one here, I think, cluster 11, which is understandable that this is a bit weird because the diffusion pseudotime just averages out these these things, and the principal curve pseudotime tries to go through it. So, and the last thing that people do is, this was, what was actually my L value was 100, I think, for these things, right? Sorry, I'm have here xm vectors to the L, 300, exactly. You can try this a bit with different L values and always look at this plot here. And when you see how the plot for too small L values, it becomes really like this. And for large L values, it becomes flatter and flatter. And of course, you want to, this dip here, this corner, you don't want it too cornery. But with L to the 300, the eigenvalues go down quite quickly, as you can see here. So essentially, the first 20 or so, the first 10 or so components should probably be enough to judge the eigenvalues. And even though we now calculated with 100, it's not so important. And with these 300 or so eigenvalues, why did it look differently on this one? Here, I think I messed up and showed you the other one. It's only here the first, it's only if you take 300 steps, then only the first 6 or 7 eigenvalues are actually relevant, and all the rest is 0, which also means that of this matrix X, only the first 5 or 6 vectors are relevant. So if we want to describe a position of the cell and calculate the distance of the cells, we describe each cell only with 5 or 6 components. So we again have done some kind of dimension reduction. And we can now take, for example, simply the first 2 eigenvalues here. The very first eigenvalue we don't take because that's the eigenvalue which is associated with the equilibrium distribution where everything is spread out completely. And if we look at it, you notice it simply puts in each vertex 1 over the degree. But these 2, they are useful, and I plot now this eigenvector against that eigenvector. And if I plot these eigenvectors against each other, then we see this plot here. First eigenvector and second eigenvector. And now you can see really nicely how we can now see an arrangement of these cells along this thing. And we see not only a linear pseudotime, but we really see the whole process sort of split down into filaments, so that in every cluster we get something like a natural ordering of how things diffuse through this cluster. Let's see if this works or if I've messed things up. Nope. I have to run this through once more. I even have a 3D plot, made a 3D plot which looks at the first 3 of them, so that you can look at that. But this is why people are actually interested in these diffusion pseudospaces, because it gives you it sharpens your view for the transitions and removes all the bulking of something else. So if you had something which in PCA space looks like this, sort of spread out, then after you do this diffusion transformation, it really shrinks down to this quasi one-dimensional branched structure, as you see here. And this is an interesting way how this, we call it the graph spectrum, shows us the rough structure of the graph. No, it now goes through here, console. Ah, okay. Yeah.
This method requires, however, finding 2 points that are sensible to start and end. So those have dissimilar distance to start.
Yes and no. If you want to use a pseudo time, but for the pseudo time, I often have it something which isn't quite noticed here in my example, because in my example we have one trajectory always, but that's not so much the typical biological case. Often in biology, you have these branching trajectories. Take the most extreme example, embryonal development. We start with a single fertilized egg cell, which gives rise to all your different body cells. So we know that the cell, when it divides, you have this 4 and 8. I'm sure you have seen at high school biology these movies where you see how an egg cell divides and divides, and then gives this blastula and the morula, and then invaginates, and then it splits into these 3 so-called germ layers, with the outer germ layer then later giving rise to the skin and the nerves and the brain, and the inner brain layer, I think, to all the gastrointestinal tract, and the middle germ layer to the other organs. And in that way, you get this branching, where you start off with a cell, it splits a while, then it branches into 3 germ layers, then maybe the outer germ layer, the ectoderm, will split into the neuronal and the skin-like cells, and so on. And there, this thing is really useful. There, if you now wanted to do this, and you had a branching stuff like this one here, I could choose this as the start and this as the end, and then assign pseudo time for these cells, while these cells would be something like this thing which goes up here. But for this last part, for this picture here, I haven't chosen a start and an end point. Here I've just used for each cell its vector, its row in X. Remember, we take T to the L, and I have chosen L equals 300, and then I calculated this matrix X here, which has 1 row per cell and 1 column per eigenvector, and the norms of the columns rapidly go down with increasing eigenvalue, because they are proportional to the eigenvalue times L, because that's multiplied here. You see here is the multiplication with lambda to the L. So the ith column in this thing has a value of its eigenvalue to the 300th power. And this is why the norms of this matrix X goes down quickly, and each cell is reasonably well described by maybe the first 6 or 7 components of L, and of which I have plotted here the first 2 components, and they already and they now give me this structure. And here's no first start and end cell. This is just whatever ended up in the first 2 components. I can now make a plot of the second to the third, which will probably then separate up this cyan triangle from the magenta triangle, and so on. This is why here I try to do this 3-dimensional. So here now I plot the first 3. And as you can see, this is how it now looks like. This is what we've seen before. And this is now the third one is split over there. And now we really have nicely split this in different things. And if you want to recognize such branching patterns, then this is a very useful projection. This is now second, third, and fourth, because the first eigenvalue is exactly 1, and you always have to skip it. This is because in a matrix process, you always have a final equilibrium distribution to which no matter where you start, you always end up in the same equilibrium distribution where everything is just randomly spread out, and that one is associated with the eigenvector, with the eigenvalue 1. This is why I skipped that one and started with 2, 3, 4, but I can also make 3, 4, 5, for example. And let's see how this now looks like. And yeah, you can play with this Unfortunately, in my web page, I couldn't put this 3D thing. There is some way of putting this in HTML, but I don't know how. But that's it. So the one thing, other questions to that part? Then, yeah, I should probably have found a data set with more branching, like the embryonal development data set, to show this a bit better, how this splits out. But I will now do something else which has even less branching, and this is going back to my grid here, my 30 by 30 grid, because I want to show you, and this has now little to do with biology, this is maybe more for the physicists in the room. Who knows about, has heard about the heat equation? Exactly. Who has noticed that we've been talking about that the whole time? Okay, 2 people. So, in the heat equation, as I said before, typically, if we look at diffusion, we typically imagine diffusion on some plane. Like, I put some spot of ink and see how it spreads out. So this grid here is maybe a good model for my plane diffusion. And this is why I made here this matrix, where I made here this vertex matrix. So you see, this is my vertices, which are now a 30 by 30 grid, and I number them like this here, 1, 2, 3, 31, and so on. And here, this is how this thing works. Now I make an adjacency matrix with 900 by 900 values. And in order to make an edge, I can do something like this here. I look up what is the vertex number of a grid point in this row, in this column, and what is the vertex number of a grid point in this row in the column next one to the right. And I put a 1 into these. And if I do this now, I get my adjacency matrix, and I can calculate the eigenspectrum of the eigenmatrix. In principle, I should now first make it, should now multiply it from the right to the left with this d to the minus 1/2, but this doesn't change the eigenvectors, only the eigenvalues. And I'm interested in the eigenvectors now, because the nice thing about the eigenvectors is now that the eigenvectors are, I showed you the thing before where I said I start at some random grid point and then watch how it diffuses outwards. But what I can also do is I just look at the eigenvectors, and each eigenvector, of course, assigns a value to every individual grid point. So I can draw it as an image. So I can, for example, take the 7th eigenvector, and now say this 7th eigenvector, I have to arrange this again in a matrix with 30 columns. And then it looks like this, and that one, I can now tell the thing I want to image. And then I get this image here. And I've done this here for the first 100 eigenvectors. And let me run this here. And here you see them. No? This is the first eigenvector, second eigenvector, third one. I always put the eigenvalue in top, so you see this is the highest eigenvalue except for the 1. And this here is now, these two have the same eigenvalue, and they are symmetric. This is the third eigen thing, comes this, and so on. Anybody seen these patterns before? Yep. Now, of course, it's on a square. Orbitals, if it was round, it would look even more like orbitals, but of course, it's a 2D case. There's one nice high school physics experiment where you see this pattern exactly. Exactly, the Chladni sound pictures. So you take, in that case, because I want a square, I would take a square piece of metal, suspend it somehow, and then I use a bow, like for a violin, and I stroke this thing, and then the thing swings. And I put fine sand onto it, and wherever it swings up and down a lot, the sand is blown away. And where it doesn't swing, in the nodes of vibration, the sand stays. And with this, you can visualize these things. And then you can also see exactly these pictures here. Yeah.
Is there a way to understand why in our matrix, which has no special structure, there are these eigenvectors? An interesting structure.
You mean this here now?
Yes.
Well, our matrix does have a special structure because the way how I made my adjacency matrix. I took the eigenspectrum of this adjacency matrix where I put a 1 whenever two elements of a matrix are neighbors, either side by side or top bottom. And in this way, I have encoded this grid here with its connection here into this thing. So basically, I wanted to describe a square plate, and I discretize the square plate by little. In physics, we always then have these string models where we say we have a couple of weights, and between each couple of weights, we put strings or something else which is elastic. And what we describe now with our matrix is, interestingly, not the 2D geometry of this thing, but we just describe for each point what are its neighbors by putting a 1 there. And if we then rearrange the points such that the neighbors are next to each other, we see these colors again. The thing which the next question you can ask is, of course, why we talked about diffusion. And this now more looks like waves. And where's the connection there? And let's see if I manage to, and maybe, what's the time? I have a short attempt to look at this. So we have our matrix A, or our matrix T, which is e to the minus 1a. And now we want to see what's happening what is happening if we wait for a small time step. And so far, we always made discrete time steps, where we always make a full step to a neighbor. But if we only wait for a very small time step, then we can imagine that on each vertex, there's amount of, and the original physical situation that people looked at it was heat. They imagined that each grid point has a certain temperature. And two neighboring grid points, they exchange temperature. When one is hotter than the other, then heat will flow from the hotter to the cooler one. And so if I want to know how does the temperature, so Ui is the temperature of this thing at some time, the temperature of grid point I. And this temperature, to some time point t plus delta t, is the temperature before plus all the heat that has flown into it. And for this, we go through all the neighbors. So let J run over all the neighbors. And there we have to now think about it. If the neighbor is hotter than our index point, then heat will flow towards us. But if the neighbor is cooler, then heat will flow away there. So we have to look at the Uj of t minus Ui of t. And with this now, we can see how the change here, the change in time here depends on the change in space here. And once we add this up, if we now add this thing up, for example, for a square grid, then what we will get is something like we have, yes, let's write this as Ui of t plus, Ui of t is always in, oh, and a delta t I've forgotten. Because of course, the longer we wait, the more heat will flow. So I have then plus delta t di Ui of t minus, so this is the plus delta t and
So now what you can see is we have here, I have to put this here, put that here. And this I can consider as a bit a difference operator. Because if we now have here our index point and here the surrounding neighbor points, when you see we take all this here minus 4 times this and plus 1 times each of this. And in a way, and this is a gradient. Sorry, this is a Laplace operator. This is a second derivative. If, for example, we take only a line where this is one-dimensional, then this would be, then what we see here is the overall change is we look at the difference here, the difference between the left neighbor and our index point. This is how much heat flows to the left. The difference between the right neighbor and our index point is how much heat we get from the right. And the difference between these two differences is how much change we get. Because if u minus 1, u naught, u1, and then we have here u minus 1 minus u naught, this is how much heat flows from, let's write it that way around, how much heat flows to the left, and u1 minus u naught, this is how much heat flows here in this direction. If I subtract the two, I see the difference between heat flowing in from one side and heat flowing out from the other side. And if you look at this is the difference of differences. So it's, if I then try to take the limit of delta t to 0, then this difference of differences will turn into a second derivative, because this is the first derivative, this is the other first derivative to the other side, and the difference between the difference here and the difference there is the second derivative. And then this thing here will turn into ui of t plus dt is ui at t plus and here with nabla squared now means take all the neighbors negative and the individual value positives times as many neighbors as you have. And in that way, we see how much the difference between outflowing and inflowing energy and how much the overall change is. And if I now divide by dt, then we get here u dot, so the time derivative of u is proportional, is some constant, or is proportional to the spatial derivative u dt. So du equals some constant times d, this should go. In 1D, it would look like this. In 2D, you have n plus d u to dy squared, and this is the heat equation. And if we replace the first derivative here with the second derivative, we get the wave equations. And for both of them, if we want to solve them, we come to the same thing. We have to solve, basically, we have to solve something which essentially encodes this Laplace operator. And in 2D space, this Laplace operator, as we've just seen, means the second derivative in spatial directions, so in x and in y direction, something like this here, d2u dy squared, and you can add a third one if you are in other directions. But in general, but if this is not a nice spatial grid, but some strange grid like our neighborhood graph, then you have to replace this Laplace operator by something which doesn't connect the thing with its grid neighbors, but with its actual graph neighbors. And if you look at this, what this actually then has to be, and this now I don't have the time to show, so I just tell it so that you've heard it, and if you see it in some other time in another connection, you will then notice that, yeah, maybe one can let it, that this is then called the graph Laplacian. Because if you imagine we now use the graph, then after we would say the vector on our graph at the time t plus dt is the vector on our graph at times t plus, and now I have to make my transition matrix T and apply it on that thing. And add to that one a bit what I get here. But what's wrong here is everything which I move away with this transition matrix here will of course be missing. So oh, sorry, now this time I put my U on the right-hand side of the transition matrix instead of the left as before. But the upshot is we have our probability vector at some time t, and t times the probability vector just multiplied from the right side gives us how much probability will move in one step. But if our step is very small, we have to multiply this with our small time dt that we wait. And in this small time, not all of the probability can vanish, but just a small part, because everything which then moves from a vertex to its neighbor is now missing in the vertex. That's why I have to add this term here with minus d, where d is our degree vector, because if a vertex has 5 neighbors, then of course for every amount dt that moves to the 5 neighbors, we have to take 5 times the amount reduced from there, so that overall the probability gives 1 again. And that then means, and if I now divide this thing by dt, then I get u of t plus dt. Let's go here. U of t plus dt, so this is my vector, minus u of t, that's this thing brought to the other side, is t minus d u of t. And with dt here I also bring on the other side, gleich dt is t minus d of ut. So here we have now this thing here, which I can also write as t minus d. I can also write this as a minus 1 times a is t to minus, no, that doesn't get right now. I've got the thing. But now you have this thing, which is the transition matrix minus the diagonal matrix, or sometimes you also see it at d minus I or a minus I, and somehow I got the normalization wrong. But the adjacency matrix minus either the identity or the degree matrix gives you something which here takes the same space as the Laplace operator in the heat equation. And this is why you call this thing here the graph Laplacian. And that's interesting because it shows you how we generalize things in math. We started off with Monsieur Fourier, who wanted to understand heat transport. So he started off with this heat equation. Equation. Wikipedia had this very nice picture for that, I think. Yes. Now something happens here. He started off with saying, imagine you have a grid, and in this grid you have ones where the red thing isn't zero otherwise, and you want to know, because you have applied heat at the red stuff, and you want to know how the heat spreads out. And in order to do this, he thought about how to make this a grid, then came to this equation, when he realized that what he's doing here is something like a second spatial derivative, and then wanted to find this out and thought, okay, I have to find the eigenmodes of this thing here. And the eigenmodes of this thing here are exactly these things here. So Fourier realized he has to write his initial condition with this crescent moon shape thing as a linear combination of these basis shapes. And if he manages that, then he can calculate the heat equation, because he can solve the heat equation, because for each of these basis shapes, we know how the heat changes. For example, if we start off with this thing, making this hot and this cold, then heat will just equilibrate slowly until everything has the same heat, and it will keep its shape. The shape of this thing will not change. And the eigenvalue here tells us the speed in which the heat goes out. If we do something like this, then it will be much faster that the heat equilibrates, only 2.8 instead of 3.7, because it has to go less space to get to this next, from the hot to this cold things. But again, it will, when you look at the heat distribution, it will slowly go down to 0. And if you then add up your initial condition as a superposition of these things, then you come, you can calculate the temporal evolution as shown by that, by that part here, by decaying the heat difference exponentially according to the eigenvalues and always adding up the individual basis function by nu. And this is, of course, Fourier analysis, and that's how this whole thing got started. Interestingly enough, you always learn it with for waves and sound, but the starting point was solving the heat expression, and only later people realized that if you go from the first to the second derivative, you can also explain things like drum heads and swinging subjects. And what I like especially is that it's been so easy to actually simulate this on a computer without going through the whole infinitesimal stuff of Fourier transformation. I get all my Fourier base functions. You can see how these are typically wave-based functions. Just from this very simple code, where I simply took a grid, made these things, and calculated the eigenvectors of it, which now also shows you how you would do that if you wanted to calculate the sound patterns of a strangely shaped drum head. You just make a grid in that shape and then go through that, and then calculate the eigenvectors that way. So, yeah, as I said, for the single cell biology part, it's only about we use this diffusion process in order to have a new way of emphasizing the flow along the elongated structures in our state space, and sort of while averaging away the stuff orthogonal to it. And for this, we use we calculate the eigenspectra of this adjacency matrix or of a transition matrix, and if we want to change this whole thing from our discrete time step into a continuous time step, we notice that we have to do this thing here, where we have to subtract the identity, simply because in a small time step, not everything moves, but only part of it. And with that, we get to the original heat equation and get back to where we all started. And yeah, you don't really have to remember all these connections. This is more something that I thought I'd tell you because it's useful, because Fourier analysis is something you see over and over in different areas of math and physics and also of scientific computing. And every time it looks quite different. But the more different perspectives you see on it, the better an understanding of the core idea behind it. And so I now offer you this as a new puzzle piece to all the other stuff you might have learned in other lectures on Fourier analysis, maybe coming now from a very different direction. And we again end up by something like this here. Okay, other questions, remarks, whatever? Then for tomorrow, for the exercise, those who've been in the last exercise, but I forgot to upload it as a written thing, I suggested that as a homework you can try to cut our autoencoder in half and take only the encoder to build a classifier and try this out on this old MNIST data set. Don't know if anybody tried that. Did anybody try that by any chance? We can try this tomorrow. The other thing that we can try is to take the, and I want to see where we can both, do already in preparation for the last lecture, to take the UMAP loss function and give it into PyTorch and see if we find something UMAP by simply writing down their loss function and just running in a general optimizer. And my expectation is it won't really look like exactly the same UMAP because there's some simplifications in the loss function, but you can write down a simple loss function where you simply say, where you only make a distinction between cells which are neighbors versus cells which are not neighbors. And well, might actually, and I tried this a year ago. And I don't know if I find my old code, but if I don't, then you have to help me tomorrow to write it. And again. So, otherwise, oh, and you all got an email, I hope, about if you want to have and take an exam or need a grade, just send me an email, and then I make a list of everybody, and then I send around a big doodle or whatever where you can try to find times when you want to take that. And yeah. I somehow hope we can keep this simple because asking 20 times the same questions gets a bit annoying. But it's probably still easier than trying to come up with some way of doing a written exam on this, which is maybe it's too complicated to ask you to program something, it's too applied to ask you to prove something, so I would have no idea how to make a written exam about this kind of stuff. But chatting a bit about it would always work. Okay, then see you tomorrow.